Hi, this is Miss Litton, and this is my excellent AP Biology multiple classes 7th period support. Say hi. Hi. All right, so we will review Chapter 6, Metabolism, Energy, and Enzymes. Today is what day? Thursday. Your quiz is not tomorrow, right? When is it on the calendar for? Wednesday, because Monday is holiday. And then Tuesday, thank you for those, my good Jewish children. Okay? Is that what holiday it is?
Yeah, it's Jewish holiday.
Oh, sorry. Thank you. Thank you. On Tuesday will be a 246 day. Wednesday is a 135 day, and that's the day you're training in your lab. Yes? Yeah. Okay. So just let's briefly touch on the lab. Okay. Okay, here we go. So on your lab map, you already did this part for sure, right? That's not a problem. Do we need to go over that? No. Okay. So then we wrote our null hypothesis, and we're going to make sure our null hypothesis states that, what? The concentration of the substrate, in this case, what was our substrate? Hydrogen peroxide, would not have any effect on the amount of oxygen produced, which is our product. So we're saying the amount of substrate does not affect the amount of product we get. Okay, that would be our null hypothesis. Okay, independent variable was what? The concentration of the hydrogen peroxide. What was the dependent variable? Yes, and I took a picture. Mina did a great job. So did many of you. I just threw it in there. Hope that was okay. On the lab map. Okay, then you were to take your data, right? And you have your group rate. Where do you get your course rate? Okay, and let's go check that. Do we know if we have some outliers? Okay, I don't have anything for group 5 and group 8 and period 3, group 3. Okay, but as we look at this, look at the 0%. Is that what we would have expected? Yeah, and I know some of you were concerned because you looked at your data and you're like, why is it at 174? But there is, in fact, oxygen in the air, right? Mm-hmm. But what we noticed is it didn't really change over time, right? If we did what we started out with and what we ended up with, and we took and divided it, that's pretty close to 0, so that looks really good. Okay, then when you looked at your 1.5% and your 3%, if you look at that, look to see at your numbers. This is concerning to me. Period 1, group 7, because there's not much of a difference right there. But what you would tend to expect, right, is whatever you got at 1.5%, it would make sense to me at 3%, you would expect it to be what? Double. So look at your data and see if it has double. You see that? Okay, that would be, so I don't know what you got, but you can look. You would think it would be that about, this is interesting right here, this 3. There's a couple in here. I don't understand this. Period 3, group 5. You got 5.7 and 8.3. You didn't do something right on that. I would call that definitely outlier data. So as we go through, we can see, though, the trend is very little oxygen generated over that time with 0%. We have 10%. And then we, as a rate per minute, and here almost double that, 23%. Do you understand the data, how the data works? Okay. Whether or not you're outlier, I'm going to have to look at that. Probably first period, we're going to go attack that data. Here. So when you did your data, the next thing you would have to do underneath your data table, this is where you would do your calculations. You would show me your rate, parts per trillion per minute, right? So you would take final minus initial, whatever you got at 3 minutes, minus what you had at 0 minutes, and then you would divide it by the change in time. The change in time is how much? 3 minutes divided by 3. That was your group rate, and then you can record the course rate later once I've gone over and gotten rid of the outliers, because you don't need that for the rest of your work. You just needed your data. And then do your chi-square test. Now, chi-square test, that's kind of hard because you're like 0, 1/2, 3%. So what I would say is, if the concentration doesn't make any difference, then the 1.5% should be the same as the what? 3%. Your 1.5% data should be the same as your 3%. So make your 3% the norm. Does that make sense? And so we do observed minus expected over expected. So at 3%, observed, whatever your rate is, minus what you expected that at 3%, divided by, right, observed minus expected. And so then you're going to end up, that number is going to be 0, right? And then when you do the other one, you do your 3%, right? Or whatever your 1.5% is minus your observed, which would be 3%. Take your difference, square it over what you expected. So you really only have one calculation to do because the other one's going to be null, right? It's going to be 0. Okay, and then look to see what's your value there. Do you have, is it, because for some, if your 1.5 and 3% numbers are really close together, then you are not going to have a significant difference. And remember, your degrees of freedom is still going to be 1. And so you're not going to have a significant difference. So at that point, you would say, no, it didn't make any difference about my concentration, especially if your two numbers are really close. But for most of you, you have a drastic difference between the 1.5 and the 3%. Are you okay? You know what to do? All right, so you're going to do your rate and your chi-square underneath your data table. Okay, then your next part is to make your claim. Do your trend based on your reaction rate, what happened. And then what's your evidence? Just give me your rates. And then support it with your chi-square, and then your reason. Why do you think it turned out that way? And then any kind of errors where you could have. Like, I would give a really good error would be, I gave you a syringe for water. It went up 1 degree. It's been like staying at 75, 76. It's not been changing. And yesterday I thought I was going to have a heat stroke. Who was in your 5th period? Were we melting? No. Oh, I was melting. Oh, yeah, I am. And so one source of error would be that I should have given you, this is not any fault of your own, but I was afraid to give you because some of you are mixing up syringes and leaving syringes switched for me for my next class. I gave you a 10 mil syringe for the water and a 5 mil syringe for the hydrogen peroxide, or a 3 mil syringe for the hydrogen peroxide. And the reason why I did that, I didn't want you to mix them up, but you couldn't get exactly 2.5 mils of water, so maybe you overshot it. Another source of error would be your error, which some people redid their experiment and it worked, was you didn't realize what a 1/2 mil was. The tiniest syringe with the catalase, a 1/2 mil was half the test tube, right? Or half the syringe. So that is something you should have corrected when you saw it. You should have just redone the lab, okay? Some of you got weird data and it was 5. Whoever's team 5. Hey, was team 5 one that I had bad data? I didn't like their data?
That was ours.
Oh, you redid it. Okay. Yeah. But what they did is they said, hey, I think it's the probeware. And they changed out the probeware and then it worked fine. So those are things you couldn't control your probeware, but we problem solved until we figured it out. We thought maybe their solutions were contaminated. We tried a couple different things on them. All right? Okay, so those could be sources of error. And then conclusions are just like what you did before. So your goal, 1 and 3, I'll see you tomorrow. You will have the whole period to work on that, and then your POGL. So I personally would do the POGL first so it's done, and then I would work on the lab. That would be the strategy. 5th period, I won't see you tomorrow, remember. 5th period, you're going to go to Ms. Breaker's classroom, which is next to Ms. Galbraith's. So bring all your stuff, your calculator, your Chromebook, so you can do all your work. Okay? Questions or concerns about tomorrow? Okay, so let's do chapter 6 now and review that real quick. Okay, is it still recording? Yes. Okay, so first thing about energy is you just want to make sure you kind of review the idea between potential energy and kinetic energy. What would food be an example of? Potential energy, and it's a subcategory of potential energy, and that would be what? Chemical energy. Good. And kinetic energy is energy of what? motion. And we talked about there's all different kinds of energy, right? Solar energy, and we're trying to get it into chemical energy, and plants do that for us. We reviewed, and you're going to want to know the difference between the two laws of thermodynamics. The first law says energy cannot be what? Created or destroyed. It can just transfer, right? The second law says every time you transfer, you lose some as heat, okay? This is why you have radiators in cars, right? Some sort of cooling thing in the car. Because as you're burning the fuel, not all of the energy is going to make the tires go round. It's just generating heat, and when you generate that heat, it could overheat your engine, so you have to have some sort of coolant in your car. All right, then the next big topic we discussed was entropy. And remember, entropy was the amount of disorder in the system. And all of our bodily kitchens are going to greater and greater disorder. And the reason why we eat food and breathe oxygen is to fight that entropy. Okay, we're always fighting our bodily entropy. So what would happen if we lose the battle? You die. Yeah, death is the final battle lost of entropy. You are not going against it anymore. So we talked about who has more energy. So we looked at a low entropy situation, has a lot of potential energy, right? This is low entropy, but a lot of potential energy. Whereas here we have high entropy, disorder, and it's low potential. So we looked at things like ice cubes having more potential energy than you would liquid water. Okay? And then here, the difference between glucose and carbon dioxide in water. Glucose is C6H12O6. You could still have 6 carbons over here and have 6 CO2 molecules, but who has more energy and why? Whoever's next to you with the darkest shirt, explain why this has more energy than carbon dioxide and water. Go ahead. So you know about carbohydrates, the 4 important organic molecules. Carbohydrates, proteins, lipids, nucleic acids. So if you were carb loading before a race, would you eat a bowl of sugar cubes or a bowl of pasta? Pasta, right? Okay? They're both, you're like sugar all the way. Ultimately, pasta is just a bowl of sugar cubes, but the sugar cubes are connected together, and because they are connected together, they are more complex and they have more potential energy in them. All right? Now, another thing we looked at, and this becomes really important for cellular respiration and photosynthesis, is when you can have a difference across a membrane. And when you have a difference across a membrane like this, you have a difference in what? Concentration. What else? Charge. What else? And pH. This would have a lower pH. This would have a higher pH. This has more hydrogen ions. This had less. This has more positive, so relative to this positive, this would be what? Negative. Okay? That is a lot of potential energy. And if we have these hydrogens go through here, one really cool enzymatic protein to have it go through is what? ATP synthase complex. Because when you do that, when you allow it to go through, then you can make ADP become ATP by snapping on a phosphate. So we learn about this potential energy because this sets us up to understand both photosynthesis and cellular respiration. Ask your friends around you, are you okay with this? Are you understanding this all right? Questions you want to ask me about that? Now, the energy of the universe is going to greater and greater disorder. So how does that make sense? How can we have an ordered planet when everything's going to greater and greater disorder? Oh, thank you. Could you say that louder?
Input of free energy from the sun.
Right. The sun's entropy we can use, we can harvest that energy in order to build molecules, and plants and autotrophs do that for us. Okay? Next, we talked about metabolism. Metabolism is all the enzyme-mediated reactions of your body. Now, could those reactions happen on their own? Sure. But it would be very, very what? Slow. Very slow. So enzymes will speed that up. And so we talked about anabolic reactions and catabolic reactions. Anabolic reactions, by definition, require the what? Input of energy, right? Catabolic reactions, what do? They but yet they still require energy. What is that energy that they require?
Energy of activation.
Boom! This girl's got it. Okay? So energy of activation is that hurdle you have to get over. Remember to make those reactions start. Okay? So we still have a hurdle either way. This is just going to be when you have an anabolic reaction. Let's go when we're talking about delta G. Okay? When you have an anabolic, that's not what I meant to say, catabolic reaction, okay, here's your starting point, here's your ending point, you have lost energy, yes? But what they don't show you in this picture is before this ball could get rolling, it had a little hurdle it had to go over, and that's your energy of activation, and that's when we tie in our enzymes, because what do enzymes do to that hurdle? They lower it, specifically. Not for all the balls rolling down the hill, just for this ball rolling down this hill. They're very specific. An endergonic reaction, usually anabolic, you're obviously going to have to input some energy. Now, what's a coupled reaction? When you take this energy that gets released right here from this catabolic reaction, and you use that energy to help you with an anabolic reaction. That is a coupled reaction. And that is what we did here. We were coupling a catabolic reaction that loses energy with an anabolic reaction that requires energy. So for us, okay, if you want to build more muscle, that's going to require what? ATP. So you're going to couple the spending of your ATP so you can build more muscle. That is a catabolic reaction. Sorry, a coupled reaction. Just talking and not thinking. Okay, ask your bio buddy if they have any questions right now. Is there anything you want to direct towards me? Are you glad this quiz isn't tomorrow?
Yes.
Okay. Anything you want to direct towards me on this? May I provide you assistance? No? All right. Got socks on. All right. So, energy currency of the cell is the ATP, so let's review that. ATP is adenosine. Look at how cute. Sorry, I like the skirt. Adenosine is a really cute tennis skirt. Adenosine triphosphate, so we have adenine right here. What is adenine? A nitrogenous base. Where are the rest of you? Be with me, 100%. You can do this. What's another, who does adenine like to bind with? Thymine. And guanine likes to bind with cytosine. Adenine, purine or pyrimidine? Purine. What's another purine? Guanine. They have how many rings? And a purine always bonds with a pyrimidine. Adenine and thymine have how many hydrogen bonds between them? 2. How many between guanine and cytosine? 3. This stuff never goes away, right? We got to keep going back and reviewing everything. But anyway, there's some adenine. There's ribose, phosphate, phosphate, phosphate. What kind of reaction is taking place right here? Input of water. Hydrolysis, hydrolysis, right? So it's snapping off one of these phosphates. Here it is, this inorganic phosphate. You're now left with ADP, but what do you get out of it? Energy. Use that energy to do some anabolic reaction, right? Use that energy for some anabolic reaction. Here's one. C and D, to become A and B, they need some energy. Where could they get their energy from? ATP. So that energy was transferred from ATP molecule to C and D so they could become what? A and B. And we looked at that. We looked at the coupling of anabolic and catabolic reactions with the building and breaking of ATP. Okay. Next, we said if glucose is, or if ATP is $1 bill, we think of glucose as $100 bill. And that's hard to spend at the dollar store, I guess, unless you buy 100 things. So we need to break down that glucose in multiple small steps in order to slowly release the energy out of it to make ATP. And why would we even want the ATP? 3 kinds of work: chemical work, transport work, and mechanical work. Chemical work, we might need to synthesize some molecule. Transport work, you know all about transport work. Yes? Nothing there looks... Oh, what's this right here? Just straight up diffusion. But here is still diffusion, but you're using a protein, so what would that be called? Facilitated diffusion. Okay? Here you're moving something against their concentration. That would be active transport. And what are you using? A what? A carrier pump. Yeah, good. All right. Then we said also mechanical work, like actin and myosin sliding past each other in a muscle contracting. You don't have to memorize that. When muscles contract. When flagella flagellates. When cilia moves, okay? That would be mechanical work. I don't think that's a good word, but we're just going to move on. Hope you're having a good day. All right, so metabolic pathways. We are a bag of chemicals, and we need to catalyze specific reactions at specific times. Catalysts can help us do that. Organic catalysts are called enzymes. What would you tell me is a key part of an enzyme? It's, thank you, perfect, active site. Now, what if something gets in here and gets in this active site? What would you call that? A competitive inhibitor, right? Why would you call it a competitive inhibitor? It's competing for the active site. If something binds somewhere else in this enzyme and makes this enzyme inactive because of its binding over here, it's not even touching this, but this shape changes as a result, that would be a what? Non-what? Competitive, right? And what's this secondary site that it's binding at right here? What would that be called? Allosteric site. Good. What would you call it if it was some sort of organic thing that had to bind to it in order to make the active site work? A coenzyme. And if it was inorganic, you would call it a cofactor, like if it was a piece of metal or something like heme and hemoglobin, iron. Is that okay? A little bit? All right. Okay, so, oh, this is important. Okay, so there's the induced fit hypothesis, which is, remember our toothpick aces, whoever you were in each of your classes? You changed your hands momentarily in order to break the toothpick, right? But you didn't stay in that position. You released the toothpick and grabbed another one and broke it again. So you had to change your shape temporarily. That's your induced fit. Just to facilitate that reaction. So you can tell if this looks like a mouth, then the mouth looks like it's closed, right? Looks like it's closed down there. So it's changing its shape in order to facilitate that reaction. I think it's here as well. We said you only need a little bit of enzyme because it can be used again and again. It is not altered by it. It is not consumed by the reaction. It will continue to work. We talked about how they're named, and they're named after their substrate. And we talked about enzymes do not change your beginning and ending level of energy. All they change is the what? Yeah, energy of activation, which it lowers that enough that the kinetic energy of the molecules themselves will overcome the smaller energy of activation. Hey, ask the person next to you, how are you doing on that? All right. It's a lot of material, huh? You can do it. All right. Factors affecting enzyme activity. This was your lab, right? Your lab was if you did a null, but what we know is that if you increase the substrate, you will increase the amount of what? Product. If you increase the substrate, you will increase the product. You will increase the reaction rate. And we looked at that. You had a reaction rates that were primarily double, right? When you calculated your reaction rate. So you increased the reaction rate when you increased the amount of substrate. We also mentioned that pH is critical because most enzymes are what? Made out of what? Proteins. Protein shape can be altered by a couple of things. One of those things is what? pH. If you change the amount of hydrogen ions surrounding it, those hydrogen ions will bind, right? They're H+, they would be attracted to anything that is what? Negative, or even partially negative. They will bind on there, then that can set a chain reaction throughout that entire molecule as it rearranges all of its bonds. Okay, and so it will change its shape, and if you change the shape, remember structure equals what? Function. Change the shape, you'll change the function, the functionality. Okay, besides enzymes affecting, what else can affect it? Temperature. So enzymes have a specific temperature they work out. By and large, though, if you increase the temperature, you make the molecules move faster, they have more inherent kinetic energy, it's more likely for the reaction to happen. Up to a what? Certain point, yeah. And then what do you call that if you overdo the heat? Denature. Should you ever change the pH? No, pH is really specific. And remember, every time you change pH, it's a change in what? 10. So if I tell you it's 75 degrees, no, just kidding, it's 74. Is that any big deal? No. But if I tell you, oh no, it's not 74, it's 64, is that a big deal? Right? And then remember, if you change it by 2, that would be 10 times 10, that's 100. So if I tell you, hey, tomorrow it's going to be 20 degrees, super, super cold, wear a lot of clothes and jackets. Oh, whoops, I meant 120. That's a big difference, right? That's why you don't mess with pH at all because of the tenfold difference each time. Temperature, it's literally 1, 2, 3 degrees. It's not as big a deal, right? Think of it like that. We looked at other things that can affect it, like cofactors, which are inorganic, that can either make it either more reactive or less reactive. We looked at coenzymes, which are organic cofactors, things like vitamins. You could phosphorylate it. You could make it active if you attached a phosphate group onto it. Then a big part, and we already talked about this, was competitive and non-competitive inhibition. And I don't want you to think that these control mechanisms are a bad thing, that no means bad. If you don't need something, you don't want that reaction to go forward, right? Only if you need it. So this is a way of giving feedback in order to maintain what? Homeostasis. So in this case, your substrate is not able to bind to the enzyme because something else is in that spot. So that would be a competitive inhibitor. And I gave you an example of that. Carbon monoxide will bind to your hemoglobin so that who can't? Oxygen. Good. And that is an irreversible. Poisons can be irreversible inhibitors where they just alter it permanently. A non-competitive inhibitor, substrate still can't bind, but when you had this non-competitive inhibitor bind to what site was that? Allosteric site. Then it changed the active site. Okay. We also talked about something that might act as an allosteric inhibitor, which is a good thing, is when you have in your metabolic pathway, you have enough stuff, whatever it is, in this case F, it can go back and bind with enzyme 1 at the beginning of your metabolic pathway and change the active site by binding in that secondary allosteric site, and then it stops building F. When the level of F gets consumed and used up, it can't act as an inhibitor anymore, and the reactions are back on. The metabolic pathway is back on. All right. All right, and still more. We talked about being oxidized and being what? Reduced. Okay. Now, When you're reduced, you really are what? More or less powerful? More. Why? Why do they call you reduced then? You're more negative because you have more what? Electrons. And electrons equal energy. So when you're reduced, you can get reduced in two ways. You could hook up with who? If you want to get reduced, who could you hook up with? Hydrogen. Because hydrogen it's not electronegative, it'll gladly give up its electron to you, right? So it will gladly give that up. So hook up with hydrogen, or another way to get reduced is to what? Yeah, get rid of oxygen. Break up with oxygen. Because when oxygen is with you, he will take your energy. So get rid of him so he does not. Okay? Conversely, if you want to be oxidized, kind of go with the name there. Want to get oxidized, hook up with oxygen and it will take your energy. Or the other way to get oxidized is to break up with your good friend hydrogen, because he would have given you that energy. And life is a series of redox reactions. Okay, so you're passing energy to one molecule to another. When one molecule, if I pass 2 electrons to her, she gets reduced and I get what? Oxidized. It's just a transfer of energy. And we looked at photosynthesis left to right. We looked at cellular respiration right to left. And we identified what happens when we see CO2 become C6H12O6. How has CO2 changed? It got reduced because it got what? It got those hydrogens. Where did those hydrogens come from? Yeah, these hydrogens right here are now these hydrogens right here. Who did those hydrogens get given to temporarily? Yes, NADP. That was the handoff. Okay, so it got given to NADP. Then these hydrogens got ripped away from who? Oxygen. So this oxygen right here, oh great, perfect, good job. This oxygen right here is this oxygen right here. Okay, it's the leftover from splitting up of that water. Okay, conversely, if we're going to do photosynthesis, let me fix this. Okay, if we're going to do photosynthesis, the glucose becomes CO2, the oxygen becomes water, the oxygen is getting reduced, the glucose is getting burned, it's getting oxidized. You're releasing the energy out of it. What are we going to do with the energy that gets released out of that glucose? We're going to use it to build what? ATP. But we're going to need some more energy, so who can capture the sun's solar energy, right? This would be the sun. Autotrophs capture the solar energy and build the glucose. Then we break down the glucose. In fact, all cells break down the glucose, right? Because who does cellular respiration? All cells. Good. All right, and we looked at a major component of that process is an electron transport chain. I probably had the middle of the room model a cell membrane for us, and we concentrate hydrogen ions on one side of the membrane. They want to go to the other side, and we make ATP. How? How did we concentrate those hydrogen ions on one side? It was an electron what? Transport. Series of redox reactions along the membrane. Some of them are powerful enough to kick a hydrogen from one side. Oh, so you didn't have to get up. You still sit there. You're just moving the hydrogens from one side to the other. What am I supposed to do? I feel uncomfortable. Okay, so we're pulling hydrogen ions from one side to the other. When they go back over, we're going to make what? ATP. Good. And we looked at it this way, right here. And I think I had a question right here. I don't know if I asked it, though. You want to try it? Go in as whatever you want. Freedom. Would it be important to bring your lab notebook tomorrow? Yes. That would be such a bummer if you did not have it. Set an alarm or something. Show the question. Oh, I did ask you this one. I remember. We can do some of the other ones if you want to. This was question 1, and here is question 2. We did do this one. I thought we had one. Okay, ready? Here we go. Oxidation is the opposite of reduction. Why can't oxidation happen to NAD+? It's already oxidized. Remember, if the light's on, it's on. You can't make it more on. If the light's off, it's off. It is involved in cellular respiration. Is it also involved in photosynthesis? Yes. Does it occur when ATP goes to ADP? No. Okay, O2, there's no neutral. You're either oxidized or you're what? Reduced. So O2 is oxygen, so it's for sure oxidized, right? Do you want any other questions on here? Or are you done? Everybody spent? Here, you can do these if you want to. If you want. Oh, do not forget your additional objectives. Your highly suggested reading and thinking. I asked very specific questions, I know, because I just did it today. So I asked very specific questions over your highly suggested reading and thinking, so make sure you review that. So here's question 1, question 2, question 3, question 4. So pause it if you need to. I'm going to go back. Guys, I'm going to try something this time. I'm going to release just, if I can, just questions, AP questions, just enzyme. And you may get the same questions again when you do your progress check, but again, but I'll try to just release the enzyme ones. And then when you do your progress check, you should get them 100% that time because you'll have seen them the second time. Does that make sense to you? So I'm going to try to do that, so look for that. I'll do it tonight, hopefully. If I don't, first period will tell me tomorrow. Or they'll wait till after class. Okay, I better stop these so you can check your answers. Most of you got that. Most of you got that one. All right. First and third, I'll see you tomorrow. Fifth, have a good weekend. All right. Make good choices.